In this video, we'll be going through DNA replication, which is a part of the biological molecules topic. If you haven't seen my previous video on nucleic acids, please give that a watch, as the content I go through in that will help you understand this video more. So, DNA replication is needed in order to make a copy of the DNA to give to daughter cells via mitosis or meiosis. It is like having the original copy of a recipe. You don't want to be handing out the original in case it gets damaged, so you give out copies. Similarly, the parent cell always has the original. DNA replication is known to occur in a semi-conservative manner, which means that the copied DNA contains one old strand from the original DNA and one new strand which is newly formed. So, how does DNA replication actually occur? These are all the steps that you need to know for your exam, but it is also a template for 6 to 10 mark questions in your exams. I know this seems like a lot of information to remember for one question, but I have purposely put everything in detail here, as even if you forget a couple of things when writing your long answer questions, there is enough to still score you 4 marks. So, DNA replication occurs within the S phase of interphase during cell division. This diagram shows you all the other stages within interphase which I go through in the cell division video. The enzyme DNA helicase breaks the hydrogen bonds between the nitrogenous bases, causing the DNA helix to unwind, which then leads to the formation of a replication fork. As you can see here, the zip represents the enzyme helicase unzipping the 2 strands, and this area here is known as the replication fork. Once the DNA has been unzipped, 2 single polynucleotide DNA strands form. These strands act as a template for the formation of a new strand using 3 DNA nucleotides. The 3 nucleotides align with the template using complementary base pairing. Hydrogen bonds form between the bases, so 2 hydrogen bonds can form between adenine and thymine, and 3 hydrogen bonds can form between cytosine and guanine. The new nucleotides are joined together by the enzyme DNA polymerase, which catalyzes the condensation reactions, forming phosphodiester bonds, creating the sugar phosphate backbone. However, DNA polymerase can only work in the 5 to 3' direction. Therefore, the 3 to 5' template strand is called the leading strand, as the DNA polymerase can act continuously in the 5 to 3 direction towards the replication fork. As you can see here, the red line shows the leading strand, acting as the template, 3 to 5. Then, the new strand forming anti-parallel in the 5 to 3 direction, which is the direction the enzyme likes. Whereas, the 5 to 3 strand is called the lagging strand, as the DNA polymerase can only work in small sections on the 3 to 5 strand, away from the replication fork, forming Okazaki fragments. For this strand, another enzyme called DNA ligase is needed to join these fragments together, forming phosphodiester bonds. So now, on to the discovery. Watson and Crick had a theory of semi-conservative DNA replication, which was later proven correct by Meselson and Stahl using bacteria and nitrogen isotopes. Bacteria are grown in agar containing the heavy nitrogen-15 isotope, as DNA contains nitrogenous bases. When the bacteria replicates, nitrogen-15 would be taken up to make their new DNA nucleotides. Then, bacteria containing only nitrogen-15 were taken out and placed in agar containing the lighter nitrogen-14. After 1 round of DNA replication, the DNA was extracted and spun in a centrifuge. As shown here in the original template, DNA with only nitrogen-15 would sink to the bottom, and new DNA containing only nitrogen-14 will rise. This is the conservative model. Whereas in the semi-conservative, all the DNA would contain both nitrogen-15 and 14, hence will settle in the middle of the tube. It is important that you understand this experiment, as they like using this experiment in DNA questions in your exams. So I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.